Good afternoon. I'm Dorothy Gray. Welcome to this news briefing from the 254th National Meeting and Exposition of the American Chemical Society in Washington, D.C. We're joined today by Mr. Michael Henley on the far end and by Dr. Michael Gower from the University of South Carolina. They've been developing a polymer implant that could help reduce weight gain and blood sugar levels. Dr. Gower.
Thanks for the introduction, and thanks for this opportunity for us to share our research with you today. So I'd like to start by just explaining a little bit about why we did this research, and then I'll pass it over to Michael Henley, who's a graduate student in the lab who carried out the experiments, and he can tell you all what he found. So obesity is a global epidemic, and the metabolic syndromes that ensue are a major cause of death and disease. And probably the most recognizable consequence of obesity is insulin resistance. And so this is usually first detected by a high fasting blood glucose measurement, and I think many of us are familiar with that. Many of us get that test yearly at our annual health screenings. And so when body tissues become insulin resistant, they can't take up glucose and use it as energy. And this not only robs the body's tissues of an important energy source, but it also results in a buildup of glucose to toxic levels in our bloodstream, a state called hyperglycemia. And so the reason that excessive body fat leads to hyperglycemia is multifactorial. But one contributing factor is that as body fat becomes enlarged, it loses its ability to help maintain insulin sensitivity in other tissues of the body. When body fat is appropriate sized and functioning properly, it actually releases factors into the bloodstream that travel to other tissues and instruct or enable those tissues to actually take blood glucose out of the bloodstream and use it. And so when the body fat becomes too large, it actually loses its ability to do this. And that's because the cells that make up the fat tissue, adipocytes, they become dysfunctional when they become overfilled with dietary fats. And so we set out to address these aberrant changes that happen in adipocytes that occur during obesity. And so our approach was actually to develop these biodegradable polymer sponges, which we have here. And we actually have SEM images of these porous structures here on the left, and you can see what size they are compared to a penny there. And so we developed these for implant into the adipose tissue for direct delivery of drugs to the fat tissue. And so I'm going to go ahead and hand it over to Michael Henley and he can tell you a little bit about the experiments that he did and what he found.
All right, so the first thing that we wanted to do was to make sure that these things were safe. And so we initially implanted them into the fat of mice, and after 14 days, we removed the tissue. But we noticed that the fat that received the polymer sponge implant was actually lower or smaller in size than those that did not receive it. And this was interesting because we're actually adding size to it by the implant. Additionally, we see that the cells, the adipocytes that he referenced earlier, were smaller as well. So because we were affecting size, we wanted to move forward and see if we could affect the function of the tissue. So indeed we were. We saw that the fat tissue that received the sponge implants produced more factors that promoted glucose uptake in tissues. And so because of that, we were very encouraged, and we thought that these would be a potential strategy for treating obesity and hyperglycemia. And so we moved towards using these in a mouse model of diet-induced obesity. So what this is just a common model where we feed up mice a high-fat diet, and over time they gain body fat as well as become hyperglycemic. So we implanted these sponges into the fat of these mice and saw that the mice that received the sponge implants gained less fat mass than those that did not receive it. Additionally, we saw that the fasting blood glucose levels of these mice were lower when compared to mice that did not receive the sponge, and that during a test where we study how the tissues uptake glucose, that the mice receiving these implants their tissues uptake glucose better than those that did not receive the implants. So to understand why this was happening, we looked at the skeletal muscle of these mice. This is because the skeletal muscle is a large tissue that takes up a lot of glucose in the body. And we saw that skeletal muscle from these mice with the sponge implants showed increased levels of a protein called GLUT transporter 4. This protein is responsible for moving glucose from the bloodstream into the tissues. And we see that this protein is upregulated in our mice that receive these sponge implants.
Yeah, so taken together, what our data shows is that by these implants that we've developed, we put them into fat and they can actually protect mice from a high-fat diet, both putting on body fat as well as increasing the mouse's ability to remove glucose from the bloodstream. And this looks like this is, one of the ways this is happening is we are affecting the molecules that are being released from the fat, which is where we're putting the implant. And so our future work is really trying to engineer these implants to really augment their effect, as well as studying them in larger animal models of human disease, which is really the next step in clinical translation. So with that, we'll hand it over for questions.
Hi, so it's Kath O'Driscoll from Chemistry and Industry magazine. I'm just interested in how this might translate to people if you were to use this idea. I mean, how many of these implants do you need to use? And I presume that's a surgical procedure, is it? And also, how long-lasting are the effects?
Yeah, so great questions. So right now it is a surgical procedure, but where we foresee this going is that these implants could be put into fat tissue subcutaneous laparoscopically. These implants last, we've studied them in the mouse up to 3 weeks where they maintain their efficacy, which is actually quite a long time for a mouse that only lives 2 years. And we do know that when we put the implants in, we have at least looked in other models and the implants last at least 100 days. So it takes a while for it to degrade, and there are ways that we could actually change the chemistry to have it last longer or actually have it degrade faster.
So you could put it in through the skin, through microneedles or something, is that the idea? And would that be the whole body that you'd need to do, or would it just be like...
So it's a fascinating question, and that's actually another area of research in the lab. So not all adipose depots are created equal, and they all have different physiological roles. So one of the questions that we have is, will this implant work as well in one fat depot relative to another? And right now we have looked at visceral fat in the mouse, so this would be like the fat that is around our vital organs. And we know it works very well when we implant it in there, and that is a surgical procedure, but mainly because we aren't laparoscopic surgeons. These could be implanted in several adipose depots in humans around the organs. But I think subcutaneous belly fat is something that we're very interested in studying in, say, like pig models.
So it's an alternative to liposuction as well. I mean, it would actually reduce the fat too, would it? It would slim people, so it acts as a slimming.
I guess yes. There's something actually fundamentally different, I would say, about our approach, though, is that instead of removing the fat, we're actually trying to reactivate its positive physiological roles. One of the things that has been found about liposuction is that backing up, the body is very designed to protect fat stores. And so if you remove fat stores one place, the body works really hard to put it back someplace else. And so there have been actually studies about compensatory effects of, like, if you remove subcutaneous fat via like liposuction, they actually can go in and non-invasively measure more fat being put around the organs, which is actually more dangerous. So we're not really looking to remove the fat like liposuction as much as we're trying to actually augment its insulin sensitivity, sensitizing physiological role.
What happens to the implants once they become saturated with the fat then as well? Are they cleared from the body in some way, or?
They are eventually, they essentially dissolve.
Thank you.
Mm-hmm.
Hi, Ben Valsler from Chemistry World magazine. I'm just wondering if you saw any evidence of immune activity at the site of the sponge. Is it possible that the effects you're seeing are actually the body upregulating in order to fight off the presence of a foreign object in its tissues?
I think we do see immune cell infiltration, and I think that there is an immune component to what we're seeing. A principal immune cell involved in the response to foreign bodies, like I guess this would be considered if you were to talk to an immunologist, is the macrophage. There's a lot of them in the adipose tissue, and we think that there probably is an interplay between macrophages and other immune cells that are being attracted to the site. And so we are working on some gain and loss of function experiments where we work with mice that don't have certain immune cells, or we deplete them to see if we actually lose or augment the effect of the implant.
I see. And with the other mice that you didn't give the sponge to, did they have a similar dummy surgical procedure so that you could check that it wasn't a response to the injury?
Want us to build that?
Yes, so we did have that control group. We called it a sham surgery, so I did the exact same procedure to those mice as I did the mice that received these sponge implants. The only thing that was different was one got the material and one got the surgery.
Excellent, thank you.
Do we have any other questions in the back of the room?
Christine Sa, American Chemical Society. Could you talk a little bit about some of the other work that you're doing related to the polymer sponge, but where you are testing resveratrol or other bioactive molecules to see if you can enhance this effect?
Yeah, absolutely. And so where we actually started with this is we got some funding from Center for Biomedical Research Excellence at the University of South Carolina to study resveratrol release from these implants. And something very interesting that we found is that our drug delivery vehicle on its own actually had some protective effects. So, you know, Michael's been working really hard on trying to understand how this works. But in parallel experiments, we are actually loading resveratrol into the sponge's matrix. And so as the sponge dissolves, and degrades, it releases resveratrol into the adipose tissue. And why we chose resveratrol is it actually can signal to adipocytes and cause a lot of the same effects that exercise can do. And so a major issue with resveratrol and many other drugs is bioavailability. We can't get very high concentrations to the tissue of interest, in this case the adipose tissue, the fat tissue, if it's eaten orally. And then there's other issues such as patient compliance and proper dosing. And so what we are finding that's pretty interesting with the resveratrol is that we can put a single implant in, release a relatively small amount of resveratrol compared to what other investigators have to feed the animals, and we're having similar, if not better, effects. And so that's kind of where we want to go. One of the avenues we want to go, when I kind of alluded to trying to engineer these matrices to be even more efficacious, delivering resveratrol and other small molecules directly to the adipose tissue is a very big interest of ours. Thanks.
And do you have any plans at this point to move into human clinical trials?
Not at this point. The next step would be, I think, pig models. They're a better model of human metabolic diseases in a lot of cases. And so we actually have collaborations starting at the University of South Carolina to work with pigs and use these implants in those models first. But certainly that's where we want to go. The major focus of the lab is developing tools that are going to translate to the clinic as quickly as possible and address gaps in human health.
Any other questions? No.
Yeah, one of the questions, I just wondered if there was a danger, would this have any effect on appetite as well? If the fat was reduced, could there be an issue there that people might want to eat more as a result? And another question was, in one of the abstracts, it mentions the possibility of reversing diabetes using these. What evidence have you got to suggest that it might actually reverse diabetes?
Absolutely. So to the first question, I think one of the things that we're interested in focusing on is what we've focused on hyperglycemia and the skeletal muscle. The skeletal muscle, as Michael said, is a major depot for glucose. It's a very metabolic organ. We're also interested in what other organs we may be having an effect on, and one of them might be the brain. One of the major ways that adipose tissue communicates with the brain is with a molecule called leptin. It's known as the hunger hormone. And so we are very interested in how putting these implants in might modulate leptin and modulate hunger and food intake of the mice. And that's something we haven't measured yet but are gearing up to do. And I think that would actually be very interesting and actually have a lot of possibilities as a therapeutic if you could actually modulate hunger with an implant to the adipose tissue. So reversing diabetes, what we mean by that is actually reversing the hyperglycemia. So type 2 diabetes, of course, is the point at which the pancreas gives out and can no longer make insulin for the patient. And so we don't know what effects the implant is having on the pancreas right now, and whether an implant into the adipose tissue could regenerate islets in the pancreas. There's a number of organs that we're studying in addition to the skeletal muscle. I alluded to the brain. The pancreas is definitely on that list, but we don't know what effects that we're having on it right now. So being able to actually reverse overt type 2 diabetes isn't so much, we think the best place for this therapy is actually in pre-diabetes. Folks that are actually hyperglycemic and with therapeutic lifestyle changes, they might also receive this implant and be better able to ameliorate their hyperglycemia and keep type 2 diabetes from ever occurring.
I think we have time for another question. Do we have any more questions? How about online? Any questions online? Well, thank you. The archived version of this session will soon be posted at bit.ly/ACSlive_DC. Please join us for our next press conference at 2 p.m. today on enabling long-distance space flights by turning astronauts' waste into plastic and nutrients. Thank you.